So awesome. Well, let's get started. Great that still so many people made it. Good to see you all for the last CMFI Mass Spec seminar this year. Yeah, it's my great pleasure to have Professor Boris Macek giving the lecture today. Maybe to give you a very brief intro. Boris did his undergrad at the University of Zagreb, and then I think did his PhD in Münster, and then a postdoc with Matthias Mann, I think first in Odense, and then at the Max Planck Institute for Biochemistry at the in Munich. And yeah, in case you don't know, so this has been, I think, really the pioneering hours of modern proteomics. I think Boris published some very important papers, perhaps some of the first using Orbitraps and HCD. So I think this must have been a very exciting time. And yeah, I think he's one of the experts in quantitative proteomics and in particular phosphoproteomics. I think he will tell us a little bit more about that shortly. So yeah, welcome, Boris. Thanks again for doing this, and I'm very much looking forward to your lecture.
Okay, thanks, Daniel, for this nice introduction, and good evening, everybody. So, as I mentioned, I have a few slides more than I usually do because Daniel asked me to give you some background on quantitative proteomics, some key methods that we're using, and also to give some examples from my research. So I'll try to do that, and I will start by presenting our group. So we are based here in Tuebingen, and we have since already 14 years now, and we have kind of three key duties here. So we do research. Obviously, at the university, we do teaching. Many of the topics that I will today mention, we also teach in form of several modules. So if you want to learn more about it, depending on the stage on what you are, you're very welcome to join these modules as well. And we also serve as the proteomics core facility for this scientific community in Tuebingen, and also to the wider scientific community in case we have capacities, right? And so we're situated here at the IFITS building, which is the Morgenstelle, very close to where Daniel is as well. And exactly, so what we do is shotgun proteomics, right? So a bit unlike you were hearing until now. So what we want to do is to obviously analyze, as usual, all proteins in a given cell, tissue, or an organism, but in order to do that, we will digest them into peptides. So this term comes from genomics, obviously, and shotgun means that we will just, fragment or digest as many proteins as we can into peptides, and we will actually analyze peptides rather than intact proteins. So this is in this review that is already 20 years old, we still have a very nice depiction of what we're really doing, what shotgun proteomics is all about. So we start with the biological sample. We will do very rough separation of this very complex protein mixture, for example, using 1D gels, so not 2D gels, but 1D gels. With pretty much just to roughly separate the mixture, to purify it a bit. And then we would cut typically 10 to 20 slices there. You can imagine that if in a typical cell line, you have about 15,000 genes that are expressed, so proteins that are present. So you may easily have 1,000 to 2,000 proteins in each of these given bands. What you then do, you do in-gel digestion. So you incubate these gel slices with a protease, and usually we use trypsin. We like to use trypsin because it's very specific, it's not very expensive, it's also very efficient, and this protease will then fragment, so digest all of these proteins into peptides, and these peptides need to be then extracted out of the gel and then analyzed by LCMS. Just a digression. So Daniel mentioned that I was with Matthias Mann. So Matthias was one of the pioneers in the field. And this simple protocol of in-gel digestion, and I'm sure you, many of you did it already, right, in order to digest your proteins and give it to mass spec for analysis, that was published originally about 25 to 30 years ago in Analytical Chemistry in that journal, and it's the most highly cited paper ever in the field of analytical chemistry, right? So this tells you that a very simple protocol can be very important for the community, and this protocol is important because it kind of married the field of analytical chemistry based on mass spec and gel-based biology, right? So we're now here. So we digested all of these proteins into peptides. And then, of course, we want to do MS, but if we would be just, ionize all of these peptides at once, we would not really be reaching very deep into the mixture. So we still need to fractionate these peptides somehow. And this is why we do LCMS, right? And LC, obviously, in our case, and I will come to that in the next slide, is a nano LC. These are very, very fine columns. We apply this mixture onto the column. We have thousands of peptides here, and ideally they will be eluted by a gradient of organic solvent one by one, ideally one by one, from the column, and then sprayed, ionized using electrosprayed and sprayed into the mass spectrometer. And in the mass spectrometer, we gather two levels of information. Obviously, we do the full scan, so MS, where then every signal is actually corresponding to a peptide that is ionized, and these peptides are then sequentially being fragmented in the second stage of the mass spectrometer and then analyzed, so the fragments then detected. And this is how we can actually get to the sequence. So from this fragmentation spectrum, we're getting to the sequence of these peptides. So obviously, we don't do this manually. This interpretation, computers do that for us, and I will come very briefly to how this is done. So I mentioned that we do and we must do actually LC, and this is just to show you how we do it, right? So we have this actually very nice and compact little UHPLC, so ultra high pressure LC, so they can go to 1,200 bar, so it's extremely high. And these are our columns here. So they are 20 to 25 centimeter in length. These are basically packed fused silica emitters. We actually pull them and pack them in our lab. So the stationary phase are those C18 beads, 1.9 micrometer in diameter. And obviously the inner diameter of that column is only 75 microns. So it's about as thin as your hair, right? So it's super thin, and we need this very small and thin chromatography, because it has an extreme, gives us extreme increase in local concentration. So a peptide, when it elutes from that particular column, has a very, very high concentration at the peak of this elution profile, and this is where electrospray gets super sensitive, right? So this is why we like to catch those peptides at the peak of their elution, and obviously this is why we use this very, very thin column. So this is the way we're getting more sensitive, so the electrospray gets more efficient and so on. Obviously, other applications require other types of columns, so different types of columns will be used for native MS and metabolomics and so on and so forth. I should also note that we actually connect this column in a very simple way using this this cross and so-called liquid junction. This is where we have to put the voltage, about 1.5 kilovolt, we put here. We do not have any pre-column or split. That means that we have a direct flow. We don't have any mixing chambers here. And obviously at the very end of the column, peptides are ionized. So this is the best chromatography that you can get, avoiding all unnecessary, you know, needles, mixing chambers, and so on and so forth. It keeps it also simple, but you are very prone to contamination. So you have to be very careful how you prepare your samples. You need to clean them up before you load them onto the MS. Then obviously, as MS, we use Orbitraps in our lab. Obviously, this is a big field. You can use many different mass spectrometers. I will not go into details. But the Orbitrap, I'm pretty sure that Daniel talked about it already. So what we really like with this particular design here is that this curved multiple basically, you know, helps to get rid of the neutrals that are then not entering the mass spec. So you have an ion beam that goes here, and then initially in the full scan, you basically put it in the C trap and then inject it into the Orbitrap. You have this axial motion that can be then very precisely measured here on the outer electrodes, gives you this axial frequency, which is then turned into M/Z. I don't go into all details right now. As you are measuring this, because you have this uninterrupted ion beam here, you have a couple of seconds to perform your up to 20 to 40 MS scans, and they're basically done. The fragmentation is done in the HCD cell, and all fragments are then moved and then measured in the Orbitrap. So you can have up to 40 MS events per one full scan. In reality, we use around 20 MS events. So why do we like these Orbitraps? This is just an example of the complexity that we have. So we have thousands of peptides after we digest our proteome. And this is just an insert of a HeLa cell digest. So if you just take the HeLa cell line, extract proteins, digest proteins, and you look in a specific M/Z range, which I have here on the x-axis, and then you do it over time, which I have here on the y-axis, you see that have extremely complicated, feature space here with a lot of features, and each of them actually is likely a peptide, right? So we need to have a mass spectrometer that can perform MS very quickly, and this is just an insert from even a very old instrument, so one of the second or third generation Orbitraps. You see that it already did quite a good job. That was probably like 15 years ago. You would see that wherever I have a cross, this is an attempted MS, and wherever I have a square, this is an identified peptide. So it already does a good job, but you see obviously features that have not been picked up. So that's why we want to have very fast mass specs. And obviously we want to have mass spectrometers that give us very high mass accuracy and resolution. And we find all of this in the Orbitrap. So I don't want to do any kind of commercial here, but really for what we do, we have very little competition here. Just lately we have this tough instruments which are combined with ion mobility that can actually compete with these Orbitraps. If I take now this same insert and if I show it in 3D space, this is actually the very same insert here shown in 3D space. You get another important feature that your mass spec needs to have, and that's the dynamic range, right? Dynamic range of detection, which basically is a value that gives you what is the difference between the highest abundant and the lowest abundant peak that you can detect either in the same scan or across the same experiment. And this dynamic range is extremely high with Orbitraps. It's about 5 to 6 orders of magnitude. And actually, talking about biological systems, if you want to analyze a proteome of a bacterial cell, you expect a dynamic range of about 6 orders of magnitude. Eukaryotic cell will have around 7 orders of magnitude. And if you go to biological samples like plasma, proteome, like tissue samples, and so on, you can have up to 11 orders of magnitude. So this is telling you that this dynamic range of detection is extremely important, and that even with the most modern LCMS instrumentation, we often need to do some additional sample preparation and fractionation in order to dig deep into the proteome. This is, of course, still just one of those peaks here zoomed in, showing you nicely how we can now look at this mass spectra in 3D space. And you see, even here, you see for each of these traces, you see 2 to 3 different, traces, which gives you this isotope cluster, and all of them we can now look at in the 3D space, and we can also then quantify these peaks in terms of their volume and not in terms of their surface as it was done before. Obviously, just to have a digression here, I know that there are bioinformaticians in the audience, so as the tool and the workhorse in our lab, we use MaxQuant for protein identification, quantification, and this is combined with Andromeda search engine in order to identify the peptides. Okay, just to recap, how do we identify peptides? Obviously, we are dependent on databases, so we typically use UniProt databases or custom-made databases if needed. And we download them, and these are FASTA files. They actually look like this, with many, many entries, and then obviously the computer then computes all theoretical, tryptic peptides and their fragmentation spectra, and pretty much matches every single acquired MSMS spectrum to many of those theoretical spectra. And of course, the higher the accuracy that you have, and this is now coming to another very, very important feature of the Orbitraps. So the higher the accuracy, the smaller the number of spectra that you need to search. So you get more specific and more accurate in the database search. And typically the accuracy that we achieve in the Orbitraps, so the raw accuracy of a calibrated instrument is about 2 ppm, and after recalibration, so during data processing, there's 2 additional steps of recalibration, which actually then put this value to about 500 ppb, parts per billion, which is extremely high, and it helps you a lot in basically in this database search. Good. So this is just to recap. So these are the instruments that we have in the lab. I should say that the Elite we don't have anymore. We gave it away. It's still a very, very good instrument, but for us it's just too slow. And we currently have HF and Exploris, and we're just applying for to get funding for a new Exploris. Each of them cost, together with the LC, about €1 million. And you see actually how the technology advanced in the past 10 years or so. So you would have typically, this is you can read, you cannot read it well, but this would be again a HeLa digest. If I take 2 microgram of that digest, right, and this would give me about 9,000 peptides within 1 hour acquisition. A few years later, we have Q-Executive HF, the very same sample, but 4 times less. So 0.5 microgram would actually almost double the number of peptides that we detect, right? And the current instrumentation with decrease in the sample amount gives us even more peptides. So this is obviously very, very important for what we need. And well, we don't expect that this will go on like this forever, but we always, every 3 to 4 years need to make sure we have the newest instrument to actually, keep being competitive, right, in this particular field. Okay, and so my job today is also to tell you something about quantitative proteomics, and this is what I will do in the next few slides. So obviously, as I already mentioned, we gather this mass spec information at 2 levels, so MS full scans and MSMS, and here in this full scan, you know, I mentioned that every peak that we see here is potentially a peptide. And obviously then we could say, well, of course, all of these intensities are different. So if I have a very intense peptide, this is very likely to be coming from a very intense protein, right? And unfortunately, this is only partially correct, right? So it's not like these intensities of those peptides directly correlate with the abundance of their corresponding proteins. And the reason is here. So the reason is that we need to ionize these peptides. We do it through electrospray in this case, and some peptides like to ionize better than the others, right? So that means that even though they're not very highly abundant, they like to ionize, and they will give you a strong response in the mass spec, and the other peptides may be abundant, right? But they, given you know, the sequence they have and so on, will not be ionizing that well. I should also say that we mostly work in the positive ion mode here for several reasons that I can discuss later. So these peptides may not want to ionize, and that basically, even though they're abundant, they will give you a low response. So this means that, you know, this fact that we cannot directly quantify out of the mass spectrum like this made people think and apply several approaches to quantify proteins using mass spec. And these are the ones that I will be discussing today. So we really like to use stable isotope labeling in proteomics, quantitative proteomics. So we want to actually you know, label both or one of or several samples that we are analyzing. We're usually comparing two, but we can compare many. We want to employ stable isotopes to label them and then to analyze them. We can do that metabolically, or we can do it chemically. And we can also use label-free quantification, so without application of any stable isotopes. And I will briefly touch upon two different methods that can be used in a label-free fashion. Good. Let's start with the stable isotope labeling. So this is something, you know, that has been employed in chemistry over many decades already. And it entered biology, really. Stable isotope labeling entered proteomics and biology in 1999, right? So about 25 years ago. And what is the idea behind it? So you want to compare two samples, right? And one of them you can leave unlabeled, right? Or you can label with some kind of agent that has naturally occurring isotopes. And the other sample you can label with the same label, but this label will contain a specific number of stable isotope atoms, right? Now, the fact is that now, after you label these two samples, you can then mix them. And the idea is that these stable isotope labels are actually identical in their chemical and physical properties. The only parameter that is different is their mass. And because you do mass spectrometry, you are able to discriminate between the light labeled or unlabeled and labeled peptides. So this is then depicted here on the right-hand side. And here you see basically the isotope cluster of the unlabeled peptide, or we call it light labeled peptide. And then after mixing, you will actually see a doublet, whereas this particular cluster comes from this particular sample. And now you see already that this allows you then to compare the intensities of these two clusters. You usually de-isotope these peaks, and then you compare the intensity of the first C12 isotope here. Obviously, this then allows you to quantify many thousands of peptides. Remember those very complicated contour plots that I showed. So then imagine if every peptide comes as a doublet, then you can actually quantify every peptide, right? And obviously, the protein that gave rise to this peptide was downregulated upon treatment in that particular sample here. Then you see something like this. You see this as downregulation. You would see this as upregulation. You would see this as induction, so only the heavy version of a particular peptide, or you see no change, right? So quantifying, labeling in that way and quantifying in that way basically allows you to quantify thousands of peptides and thereby proteins in a single LCMS run. Now compare this with Western blots, right, or something like this, where you can do one at a time. And then obviously you can quickly realize how, revolutionary this was actually when it was invented, this approach about 25 years ago. The isotopes that we use there are stable, so they're not radioactive. This is always something we need to emphasize. So they're perfectly safe to work with. These are the usual isotopes. So deuterium, carbon C13, nitrogen 15N, or oxygen 18O. And basically, they can be introduced into the peptide and protein in several different ways. This is depicted here. So how do we introduce them? So we can do chemical labeling, and I will talk about SILI. So we are introducing these isotopes already at the level of the cell culture just before we perform the experiment, and this allows us to mix those light and heavy labeled samples very, very early on in the experiment, which is very important for quantitative accuracy. We can introduce chemical modification in several ways. However, this chemical modification, in order to do that, we first need to lyse the cells, which we do separately for two samples that we want to compare. Then we need to extract proteins. So we have here, and these labels can either be introduced at the protein level or, which is more common, at the peptide level, in which case we still need to include one digestion step here. And then relatively late, we are mixing the labels. That means that all this handling before we actually mix the labels can introduce some additional quantification. We can also work with internal peptide standards, but I will not go today into this. So I'll be discussing metabolic labeling with SILI as an example, and chemical labeling with TMT as an example. So what is SILI? So SILI stands for Stable Isotope Labeling with Amino Acids in Cell Culture. It was originally published exactly 20 years ago, and it's a super simple concept. So you start with a cell line, let's say a HeLa cell line, which is a normal medium, and you split this cell line into two dishes, right? In one dish, you have a medium in which you have a normal amino acid. Usually, we like to use lysine and arginine for labeling. So here you will have normal arginine and lysine with naturally occurring isotopes of all elements. And in the other dish, you will have the same medium, everything identical, just the arginine and lysine will be in their heavy form. So a certain number of carbon and nitrogen atoms will be exchanged from the naturally occurring isotopes into their stable isotopes. Now, of course, you let the cell divide, right? The cells cannot tell the difference, right? So the stable isotope label amino acid is actually in its chemical properties identical to the unlabeled amino acid. So the cell doesn't tell the difference. The cells are usually happy, and then they divide. And with each division, they incorporate more and more, so 50% more of that label. And you see that here, and you actually see that if you would now measure the mass of any peptide, you see that in this case, gradually, this peptide turns from its light form, light mass, into the heavy form, and this heavy form gradually overtakes and becomes exclusively present in the heavy labeled sample, whereas in the light labeled sample, you still have the normal light mass of the peptide. In other words, all lysines and arginines are exchanged after 5 to 6 cell divisions in this kind of heavy labeled cell. So this is the principle of SILI when you have full labeling. So you then, of course, do a short mass spec run and you check the incorporation. You have the full labeling. You can perform your experiment, and directly afterwards you can mix those samples. It basically analyze them exactly as I mentioned, obtaining these doublets, which allow you to quantify every single peptide that you see. By the way, I mentioned that we like to label with lysine and arginine, and that is, of course, because we are using trypsin to digest the proteins, and trypsin cleaves after lysine and arginine. That means that every peptide that we have, every peptide will have at its C terminus either lysine or arginine and will be quantifiable, right? Imagine if we would use some other amino acid, only those amino acids that are present in the peptides would actually be quantifiable, right? So, or in the other words, only those peptides that contain the amino acid that you use for labeling. So in this case, we actually can label everything, and this tremendously increases the the number of data points that we have in each experiment. So what do you need, right? For SILEC, you need the growth media that lack SILEC labeling amino acids. So these are custom-made media. This makes this experiment a bit more expensive than normal cell culture. You need to have stable isotope labeled amino acids. So we have several versions of arginine and lysine that we can use in combination. So you can typically compare 3 states in one experiment. So this is a kind of maximal multiplexing that you obtain by SILEC. And then, of course, you need to work with specific growth supplements. For example, you should not be using normal serum, but dialyzed serum, because in the normal serum, you have free amino acids that are not labeled, and they would be scrambling your label and biasing your results. But this is a very, very, very established method and protocol also in our group. Obviously, it comes with some major limitations, right? So in order to perform this method, the cell or organism of interest must be auxotroph for the amino acid that you use for labeling. Now, if your cell line or your bacterium can produce its own lysine and arginine under those conditions, that of course will influence your experiment. So we cannot apply it to plants, for example. We cannot apply it to many autotrophic bacteria. We cannot apply it to many bacteria, right? Unless we shut down arginine or lysine metabolism or biosynthesis. And but we can apply it to most of the cell lines that we have in culture, like HeLa and so on and so forth. And a lot of biology is done that way. Obviously, if we cannot do metabolic labeling, then we can do chemical labeling, right? And this is just also from an old review. Various reagents that you can actually use to introduce stable isotopes into your peptide. And obviously, this is just to show you the complexity. Most of those are not used routinely, right? But also tells you that what are the most, commonly used reactive groups, right? Obviously, lysines and cysteines, right? And the N-termini are usually targeted, but several reagents. So these reagents would be synthesized typically in their light and heavy version, right? And obviously, this light and heavy version would be then mixed together, and then you would be able to obtain a doublet, right? And to analyze and to quantify the peptide of interest. Mostly they're used at the level of peptides. So as I already mentioned, you need to first isolate, digest the protein, and only then label the peptide. Another point to think about here, those protocols are quite robust, but these are chemical protocols, and different reagents will have different efficiencies. So you always need to check, obviously, whether your sample is fully labeled before you perform the mixing and the final quantification. Good. So I mentioned TMT as a means of chemical labeling. TMT is one of the reagents that targets primarily the N-termini of peptides. And this is actually from the original publication that I already noticed. It's like almost 20 years, 20 years old. So at that time it was not called TMT, it was mostly called iTrack. The principle, however, is the same. So what you have is a compound here that has pretty much two major parts. You have an isobaric tag, so you tag your peptide with this tag, and this tag consists of a reporter and the balance group, right? And it's attached to the peptides through this peptide reactive group. And what is actually interesting is that this both reporter and balance can come in different flavors, so you can have different masses of the reporter, and they are always balanced with different masses of the balance group, and they always give you a tag of a total mass of 145. So the initial iTrack or TMT reagents were actually designed for 4 samples, right? And so you would then be able to compare 4 different samples in this case. And this is how those groups were designed. So you would have the reporters, 114, 15, 16, 17. They're balanced with those balance groups, 31, 30, 29, 28. So in each case, you have exactly the same mass. So we say that these peptides, which are labeled with these different tags, are isobaric. They have the same mass. So when you mix the samples, they will all collapse into one isotope cluster. You don't have any doublets as before, or triplets or quadruplets, which is great actually, because all of these doublets, they increase your complexity at the MS level, and at the MS level, and I mentioned that we need additional speed in order to deal with this complexity. Now, they will be collapsing into one signal, and then you need to do MS in order to quantify them. So this is actually very interesting. So this group here between the reporter and balance is very labeled. So in the MS it will break apart. And what you actually have is the situation like here. Again, that's from the original publication. You have the precursor ion. This precursor ion now has all of those 4 ions collapsed in the same cluster. You fragment that, and obviously you are then getting this kind of spectrum. You have B and Y ions that give you the sequence, of course, of the peptide, and in this low mass region, you have reporters, and basically the relative intensities of these reporters tell you what is the relative intensity of the given peptide and protein in these 4 samples, right? And that, of course, is a very clever principle of labeling and quantification. Nowadays, it allows you to quantify even up to 18 samples. So it's tremendous multiplexing. However, you need to work with very high resolution mass spectrometers that allow you to resolve these peaks in the low mass region. Another problem is, of course, the fact that in order to be very accurate in quantification, you need to select very accurately every ion that you fragment. If you select several ions, of course, this biases your quantification. And if you work with a very complex peptide mixture, this is actually something that can happen, right? And you should be always aware of the potential issues. Okay, this is now just a brief, to briefly mention metabolic and chemical methods. I think that each of them has some pros and cons, right? And I will not go into this further. Daniel also asked me to mention the label-free quantification. Sorry, this slide is very busy. Actually, it was supposed to be animated. So in the label-free quantification, when we cannot or do not want to do any kind of stable isotope labeling, we can actually just measure our sample, right? So, and we, for example, want to compare cells in the sample A and B, right? So we would measure them separately without any mixing. After digestion, we're getting this a very complicated trace, so we call it total ion chromatogram here, here, and here. And we can then analyze the data with numerous pieces of software to then estimate the amounts and quantities out of those traces here mostly, right? So this is how it looks. You can do two kinds of things. Actually, do many kinds of things, right? But most commonly used are either spectral counting or extracted ion chromatogram. I'll start with the latter. So the extracted ion chromatogram, pretty much you can imagine that every peptide that you analyze here will have some kind of trace over retention time here as it elutes, right? And the software actually basically calculates the surface under the area here and in the sample A and also in the sample B, and then compares the two, right? So that's very intuitive, and obviously in this sample you would have less of that trace. And bear in mind, we do shotgun proteomics, so we usually identify more than one peptide per protein, right? 2 or 10 or maybe even 50 if it's a big abundant protein. So all of them should actually point roughly to the same ratio, right? And then additional method is spectral counting, which is even simpler than that. So you can imagine that over this retention time, each peptide will be sampled many times. So you will have many MSMS events here, right? And of course, if you have basically a higher trace and longer elution, you will have more MSMS events than if you have a less abundant peak. So just by counting the spectra, as you see here, in this case I have 4 and here I have, I don't know, 10, we can estimate the abundance of proteins. This, of course, is very nice because we don't need to label samples. This is often done when we have very low amounts or clinical samples and so on. But we should always bear in mind that the quantification is not so robust as when we use stable isotopes, and that for every sample we need to do a dedicated LC-MS run. So you need to have a lot of instrument time to do it, and you need to do a lot of replicates in order to be statistically significant, right? Okay, this is as much as I will say about theory, but I just realized that I'm running out of time, right? So actually, maybe I give you just one. And I was supposed to do phosphoproteomics. So then let me then give you an example of the phosphoproteomics that includes one of those methods that I just discussed here. So in this case, this will be SILI, right? And this is actually a project that we have in the group. So we are a mass spec-based groups, but we are biologists, and we like to use this technology in order to address biological problems, right? And one of our interests for a long time in the group are modifications of proteins, so post-translational modifications, especially in bacteria, where, 20 years back, people thought there are no modifications in bacteria, but now we know that there are many. And we are in particular interested in the serine, threonine, and tyrosine phosphorylation of bacterial proteins. And this actually led us to the field of persistence, where it's known that a serine threonine kinase is very important for the phenotype that is observed there. And what is there? So the persisters are bacterial cells that actually can avoid the action of antibiotics, but not because they become resistant, but because they shut down their metabolism. So they go into some dormant-like state, right? They kind of sleep until the antibiotic is gone. And obviously so you see them here, so the sensitive population dies. Resistors would be growing again, but persisters, they initially die out, and very low number of cells survive because they shut down their metabolism. And this is a very obviously important process because it's biomedically relevant, right? It's the cause of many of those clinical infections and reinfections and so on. And it was for a long time very, you know, obscure. People didn't really know how this is all regulated, right? And interestingly, the very first gene that was related to persistent is so-called Heap A. This is one of the many toxin-antitoxin systems. Here, I have it here, the operon. And Heap A is a kinase, right? It's a toxin because it actually stops the growth of the cell. And it's a kinase. It's a serine-threonine kinase that phosphorylates a tRNA synthetase in the active site. And it stops the activity of the synthetase, stops protein synthesis, and then you have the accumulation of uncharged tRNAs, induction of so-called stringent response, and this is believed to be the induction of persistence. I don't go really into details because I don't have time. This is counteracted by Heap B, which basically binds to Heap A when everything is normal, and it prevents its function, right? And then obviously what was very interesting that this is a kinase, and this kinase is also conserved in many of bacterial species and also many of the relevant pathogens. So we actually went in and we were asking a simple question. Is GLTX the only substrate that Heap A has, right? So how did we design the experiment? We did triple SILAC experiment. We have 3 cultures of E. coli, one grown in the light, medium, and heavy SILAC medium, right? So with different versions of lysine in this case. So we don't use arginine when we work with bacteria, it's lysine only. Triple SILAC. So we induced Heap A and we sampled them at different time points. And obviously, what we then did, we digested the proteins with 2 proteases, chymotrypsin and lyse-C. Lyse-C, obviously, because we are labeling with lysine only, so we don't use trypsin here, but the protease that cleaves after lyse-C. And then we do 2 things. We measure the proteome in many fractions to get the good coverage of the proteome, but we also, out of this digest here, very complicated digest, we incubated with beads, titanium beads, that are very specific for this phosphorylation groups. So we are actually fishing out peptides, phosphopeptides rather than proteins, right? And we are enriching for these phosphopeptides. And this is, by the way, the basics of the phosphoproteomics, right? Everything else is pretty much the same. After you enrich for these peptides out of this complex mixture, you analyze them by mass spec and then you quantify them. But you can actually detect them because you enrich for them, right? That's the key. And also when you do other modifications like acetylation, ubiquitination, and so on, more or less in every of those applications, you have some kind of enrichment at the peptide level. And that's the key for the phosphoproteomics. And here we did it, of course, after mixing these samples, and we immediately had the quantitative readout. So we can, this basically looks like that, right? That's the growth curve. We first induced Heap A. And then after 3 hours of induction, every dot that we see is a quantified phosphopeptide. And obviously those that are increasing in response to overexpression of Heap A are potential substrates, right? And very nicely, one of the major substrate is the GLTX, so the already known substrate. So this validates the method, right? But there were many others, right, that were completely unknown before, and we basically detected them here. And when we then overexpress Heap B, those that are Heap A targets have to go back, right? So we need to have some kind of trace like this. So this helped us to kind of stratify the targets. So many proteins that were involved in translation and metabolic processes of DNA and so on were detected here, and then later basically validated by Maya here, was a PhD student at the time, like with in vitro kinases and so on. So this is a principle of phosphoproteomics and application of phosphoproteomics, and we are applying this methodology to look in bacterial cells, in cancer cells, in signal transduction, and so on. And we published on that, so you're very welcome to look at our papers. This is just an example, right? And I will actually conclude with this example because I still see that I have 10 minutes, of which I probably can speak 3 to 4. And I will just conclude with this. And because I really always think that it was such a cool idea. And so how do we use now this stable isotope labeling, metabolic labeling, to actually try to study protein dynamics in those persister cells, right? Remember, it's very few of those cells, right? And it was very much unknown what happens when a cell is a kind of dormant-like state, and what also happens when the culture wakes up for persistence, right? Can we use this metabolic labeling to look at these processes? So we designed the experiment like this, right? You grow the culture until the mid-log. Then you induce Heap A, the culture stops to grow. Then you put antibiotics, so you kill everything that is not persisting. So after a long time, basically whatever survives here must be persisting, right? And then, of course, if we would just do proteomics here, it would be very difficult because you have very few living cells swimming in the pool of dead cells, right? So obviously you do proteomics and you don't know whether the protein comes from the dead cell or from the living cell, right? So the idea was then, let's use the Heap B, which we can then induce to resuscitate the culture. So by Heap B, we stop the Heap A activity. The culture then starts to grow again. And at the same time, we would actually pulse this culture with the stable isotope label lysine 8, right? So all living cells will actually start to grow and to incorporate lysine 8, whereas dead cells will not be able to incorporate lysine 8. So all of what we then need to do is just to sample this culture across the curve here in many time points, and basically look at the proteins that are incorporating the heavy label, right? For this, we don't need to have full labeling, right? We just need to look what are the proteins that are incorporating the label. This approach, by the way, is called dynamic SILI. Good, and then from the point of view of mass spectrometrist, right? And it's super simple, right? It's just basically measuring these fractions pretty much, and for every peptide that we detect in the heavy form, we are actually assembling the curves like this, right? Which tell us what is the kind of turnover, right, of that particular protein during regrowth, right, of the culture. So we're basically looking at this heavy label and how quickly it's incorporated, right? And obviously, you know, at the level of the whole culture, you see over time, we actually see this very nice incorporation. Obviously here, the cells start to really divide. We have exponential growth here, and we're saturating this at about 95%. But you see that we can now measure this incorporation very early on after we induce resuscitation. And this actually, I mean, this is just to tell us that we did it in replicates and this was reproducible. And then we can then see for hundreds of proteins, right? What are the proteins what are being synthesized when we induce the culture and when we induce this resuscitation, right? I don't go into the details, of course. We can also see what are the very first proteins that we see that are synthesized. So we still don't know what they are really doing, but we now, of course, can report that they are being synthesized very, very early on after induction. And I should say, so we're happy to see Heap B, because we are inducing Heap B, so it needs to be one of the highly synthesized at that particular point. And then we also further asked the question, can we do the same in persisting cells, right, during dormancy? And we basically just repeated the experiment and pulsed this lysine just, in the middle of this dormancy here, collected fractions. And yet again, we see much less of this incorporation. But even in a dormant-like cell, we see protein turnover. So pretty much the protein synthesis goes on. It's much it's super reduced, right? But it goes on. And interestingly, we see the complete ribosome. So all ribosomal proteins being synthesized at very, very slow speed, but they are turned over. And what was also very interesting, I don't go into details about, so we discovered also some new biology here. So some factors which are needed to, which these hibernating ribosomes are using actually, or the cell is using to inactivate ribosome. We see that some of them have much higher turnover than the others, so they must also be more important in that process. So all of this was only possible because we used this metabolic labeling by stable isotopes in cell culture, right, which also works very well in some of the bacterial systems like E. coli, not in every, but in some of them. And this is now my second part, which would be for my second hour of presentation. But as I already mentioned, I can just skip over. This was just for the bioinformaticians here, talking about some proteogenomics, which I'll be happy to tell you about maybe next year in the same seminar series, right? And obviously, I want to thank the group. I don't know who is listening, but we are always looking for master students, PhD students, postdocs, and whatever. And if you're interested and so on, I'll be happy to talk to you, right? And that's it. Thanks a lot. And I hope we still have 2 minutes for questions.
Awesome. Well, thanks so much, Boris, for this very exciting talk, both on the basics as well as your super exciting research. And yeah, for sure, we should have you again here in the new year to hear part 2. So I'm very much looking forward for that. But yeah, till then, let's open it up for discussions. I guess we still have a couple of minutes. So yeah, floor is open. Ask Boris your questions. Oh, and to those of you who leave before, because I think I'm also going to turn off the recording in a minute. Yeah, it was great seeing you all, and have a good rest of the year and a good start into the new one. All right.